Yeah, so I'm going to be talking a bit about our work today, looking at using RNA therapeutics to enhance pediatric immunotherapy. Just like to acknowledge the traditional custodians of the land in which we're meeting today and acknowledge that we're coming together from all around Australia. So sarcoma is the cancer that we're focused on in my research group. I work at the Kids Research Institute over here in Perth, and we're very much focused, therefore, on pediatric cancer types. Sarcoma is the third most common pediatric cancer, and in kids, it's largely found in the bones, so osteo and Ewing sarcoma, or in the connective tissue. So in kids, that's really the long muscles of the arms and the legs. So these are called rhabdomyosarcomas. And 75 kids in Australia each year will be diagnosed with pediatric sarcoma. And really, the therapies that we've used still haven't changed in the last 20 to 30 years. So it's still combinations of radiotherapy, chemotherapy, and surgery. Sarcoma being a solid cancer, most kids will come in and have a surgery to try and remove a lump somewhere on their body. But all of these therapies come with their own associated risks and side effects. So for radiation therapy, that's the risk of secondary cancers, the short-term and long-term effects on children's development that are very well known now with chemotherapy. And with surgery, there's the risk of loss of mobility and function, and also a risk of recurrence of disease where the surgeon can't remove all of the cancer at the time of surgery. So that's about a 20 to 30% risk of disease recurrence. And so all this means that 1 in 3 kids will still die from their sarcoma, and we really need to develop some new and improved, safer and more effective therapies for these kids. So immunotherapy is really the focus of my lab group. It's really become what we call the fourth pillar of cancer therapy in a number of adult cancers. I'm just showing here some data from a meta-analysis of some melanoma trials compared to melanoma immunotherapy trials compared to what was at the time the current standard of care with targeted inhibitors. And you can see the jump really in overall survival that was able to be achieved with the combination of these two checkpoint blockade immunotherapies. This is even a bit outdated now, so the results might be even improved. But in this study, they were showing overall survival from first-line treatment with a 5-year survival greater than 52%. So more than half the people going on to survive more than 5 years with this really aggressive and what was hard to treat form of cancer. However, these successes haven't translated over into every cancer type. And when we look at sarcoma, first of all, in adults, we see that response rates to this same combination of checkpoint blockade are down about 15–16%. And this is partly because sarcomas are a really heterogeneous and mixed group of cancers that arise from a whole different number of tissues. And so because of this, and it was noted in one of these trials that the responses that they were seeing were very highly subtype specific. And so there's not really a consistent outcome for these patients, given that their cancers are all arising from different tissues of origin. In the pediatric setting, and really there aren't a lot of pediatric trials for immunotherapy and sarcoma at the moment, but one that was completed recently in 2022 showed less than 4% of patients having a clinically significant response with that same combination of checkpoint blockade. So we haven't really been able to translate that benefit that we've seen in some of the adult cancers through to our pediatric patients. And that can be because their cancers are quite different in terms of their immunogenicity, and also because their immune systems aren't yet as developed as they might be in an adult. And so response rates to immunotherapy remain really limited, and many pediatric trials therefore still focus on those traditional therapies. So we need to really ask the question, how can we translate this benefit through to our pediatric patients as well? And so my group has been really interested in what drives response to immunotherapy on a very basic level. And so we developed these really nice bilateral tumour models where we implanted tumours on both sides of a mouse, treated that mouse systemically, and then tracked the response of the two separate tumours. And we could see in a number of models that these responded symmetrically. So the tumour on the left side would respond the same as the tumour on the right side, which means we could then resect out one of those tumours at different time points during therapy and use it as a mirror for the other tumour that was left behind to understand what was going on. And that allowed us to really classify our mice into responding mice and non-responding mice and have a look at the signaling and immune cells that were coming into the tumours and really understand what were some of the drivers of response in this setting in response to immunotherapy. And so this, out of this data really came some targets and a lot of understanding about what was skewing the certain tumours towards responding to immunotherapy while others would go on and not respond. And so we wanted to ask how might we actually target this signaling that we were seeing in the tumour microenvironment. And we came up with this idea that in solid cancers like sarcomas, that surgery was really a window of opportunity for us to target that tumour microenvironment. So we know most of our patients are going to come in, they're going to have surgery to have a solid tumour removed somewhere on their body. We also know that surgery is a very immunologically active event. And so we think that we can use this window to really target that interface between the immune system and any cancer cells that might be left behind after the surgery to really prevent those cases of recurrence and relapse disease, but also to trigger a systemic immune response that can also be effective against metastatic or systemic disease as well. And this is just a diagram showing, what we would like to do. We would like to send the right messages and signals to this tumour microenvironment to turn it from one that's weakly immunogenic into one that's more activated and infiltrated with immune cells. So to do this, the group has developed a really novel approach to therapy, which is to use immunotherapy-loaded biodegradable gels. So these are biodegradable and biocompatible gels, hydrogels, which we can use for intraoperative delivery of drugs, so during the surgical moment. And we develop them with tunable release kinetics and physical properties so they would be suited to different surgical environments. You can just see here in the panel, we tested a whole different range of gels to really come up with a Goldilocks a gel with Goldilocks physical characteristics that were suited for surgery, and we apply them in our mouse models of incomplete surgical resection, where we're actually able to remove part of the tumour, retain that for analysis, implant our hydrogel, which is full of immunotherapy, and then close that wound and follow up the mouse to see whether it's a responder or a non-responder to treatment. This is just what that gel looks like down a microscope. You can imagine these pores are forming between the different polymers of the gel as they form this matrix, and that allows us to incorporate drugs and biomarkers inside the gel. This is broken down naturally over time by enzymes which are naturally found in the wound healing environment, and the drugs are then released. And so we end up with this biodegradable gel that's safe for surgery. We can tune it for an extended drug release profile, and we can also include things like biomarkers of response. And you can see the drug release profile here of a fluorescently labeled drug. This is live imaging in mice over 14 days, and we have a good depot of drug being released over that period. And so we've used this and published on this model now for testing intraoperative immunotherapy in preclinical sarcoma models. But where I really want to take this now, and the focus of the Cure Cancer grant that I have, is to look at using messenger RNA or mRNA as a way to start signaling to the immune system to improve the immune response that we see in this setting of wound healing and residual tumour. And so we know that the tumour microenvironment is a very complex signalling environment. You've got lots of different immune cells, cancer cells, stromal cells, and they're all signalling back and forth. And so what we really want to do is use what we've learned about this signalling and then use mRNA as signalling messengers to really start influencing some of the signalling that's going on in this environment. And so this was just I guess, one of the figures I put in the Cure Cancer grant, but I think it nicely shows what we're trying to do, or the hypothesis here is that we can use surgical application of gel-based mRNA therapy to modulate the tumour microenvironment by the expression of functional therapeutic mRNA that code for immunostimulatory proteins, and that this can start to change that tumor microenvironment from one that's poorly immunogenic into one that's more immune activated and full of immune cells with better anti-tumor effective functions. So we're talking about combining kind of two novel kinds of therapies, mRNA therapy and then this gel-based delivery that we have. And we think this is a really good marriage of these two technologies, because some of the problems with mRNA therapies is that these agents are rapidly degraded by the body, and targeting and delivery to the site of action is a real issue for mRNA therapies currently in development. And so that leads to problems with achieving therapeutic dose and off-target toxicity, which is some of we believe some of these issues can be sort of mitigated by our local delivery method with our biodegradable gel. So what we've been doing is setting up assays for screening mRNA candidates. So again, these are messenger RNA that encode for functional proteins that are having a role in an immune signaling pathway or an immune stimulating pathway. And basically what we do is we have reporter cell lines and also now bone marrow derived macrophage and dendritic cell cultures that we culture over time. We transfect them with the various mRNA, and then we apply a low level of immune stimulus to those cells, and we basically look at the response. The idea being that we're trying to achieve a response in terms of the phenotype of the response that we want, or we're trying to sensitize those cells to respond to a lower level of stimulus. And we can do that by looking at protein with flow cytometry and Western blots. We can look at some of our cell-based reporter assays where we actually look at the signaling of different immune signaling pathways. We can look at the level of mRNA expression in the cell through things like qPCR, or we can look at the cells down the microscope with some of our reporter models. And so we've been using this screening assay, and we can show that at least in a dish, we can nicely get cancer cells. These are mouse sarcoma cells, rhabdomyosarcomas, and mouse macrophage cells here, RAW264, to take up mRNA and very actively produce the protein that it encodes. Here it's just the reporter protein GFP, so we can see the cells lighting up green, starting off at about 6 hours, and that expression lasts quite nicely out to 48 hours in culture, and that's just quantified there on the right. We then went and chose from the literature and from our previous work a whole bunch of mRNA candidates. I've just called them candidate sort of 1 to 11 here, which were involved in immune stimulating pathways. And we just showed that we could get transfection. We confirmed that we could get transfection of these mRNA and expression of the protein. This is in human kidney cell lines, so HEC293 cells, just to prove that the mRNA candidates could be well expressed in culture in this cell line. We then went on and applied this screening assay where we have these reporter cells. This is a HEC dual cell line that basically lights up when it activates a whole bunch of interferon stimulated genes within its DNA. And so this is a marker of activation of like interferon signaling. And so what we're doing is we're transfecting these HEC cells with our mRNA candidates, and then we're coming in 24 hours later and giving them a low level of stimulus. In this case, we're using a TLR agonist, poly-IC, and we're measuring the amount of activation of this pathway. And you can see what I'm reporting here is relative sensitization, or the strength of the signal compared to stimulated but untransfected cells. And you can see that we get a nice sensitization really of the cells to signaling when we transfect them with the mRNA. And we have a number of candidates popping up here, which we're interested in following up. So we took our first candidate, mRNA-001 in the blue there, and we thought, well, this is clearly our best candidate, but maybe it would work well in combination with some of our other mRNA. And so we just applied a screen where we transfected all the cells with mRNA-001, and then with each of the other mRNAs singly, so mRNA-001 plus 002 or 003, etc. And we found really, really strong synergy. If I just flick back there, you'll see that the level of sensitization here is about 100 to 200 fold with mRNA-001 alone. And here, when we apply 001 and 002 together, we can get up to 2,000 times sensitization. So really strong proof that we're able to, using these mRNA messengers, we're really able to activate this immune signaling at a much higher level or at a much greater sensitivity in this cell line. It was interesting, we didn't really see a lot of other things coming up in that first combination screen, but not to worry. When we apply a different kind of stimulus, here we're stimulating the cells directly with interferon beta, which acts to a slightly different pathway than the previous stimulus, poly-IC. We can see a different mRNA coming up, and this is indeed because this mRNA 004 encodes for a protein that's far more active in that interferon signaling pathway. So what we're doing now is to push forward with a second round of mRNA candidates targeting some novel and new pathways that we think can also that could also work well in combination. And we're going to do some more confirmation studies in our bone marrow cells before we head into preclinical studies. So we have some FLT3 ligand differentiated bone marrow DCs that we've been using. We've been making these in the lab and transfecting them with our mRNA as well. In a similar way, transfect with our mRNA candidate. This again is mRNA 001, our lead candidate, followed by that same stimulation with poly-IC. I'm just choosing sort of one point in the titration here to blow up for you. And here we're looking at the expression of some co-stimulatory and activation markers on those bone marrow-derived dendritic cells. And you can see with transfection, compared to the untransfected stimulated cells, you get a nice increase in the expression of these markers, which is a good indication that what we were seeing in those cell lines is actually leading to a beneficial effect in a relevant cell type in these bone marrow-derived cells. And this should allow us to look a bit more at functional changes in like cytokine production or receptor expression in these primary cells.
Can you hear me okay?
Yep.
Just to know, you've got one minute to go, if that's okay.
Yep. I'll just skip through that. Just to say that we're moving on with the in vivo delivery of these agents in our mice, and so we can inject mRNA into tumors and track that over time, up to 72 hours here. This is just with at the moment with injection. It's not with the gel, but this allows us to look at what cells in the tumor microenvironment are taking up the mRNA. It's taken up both by immune cells and tumor cells, so that's quite interesting. And we can break that down into the different subsets a little bit more. So this is the journey that we're on at the moment. And where are we at the moment? Well, we've really gone through a lot of cell-based screening now to choose our lead candidates and started the in vivo validation. We're going to be looking at encapsulating some of our lead mRNAs into LNPs now, so we can start testing them in our cancer models. And that is all from me. Thank you very much.